This conference will now be recorded.
Hello everyone. We are starting a new chapter which is biomolecules. So as the name suggests, first you have to know what is biomolecules, and related to this terminology, we have another branch of chemistry which is biochemistry. So what is biomolecules and biochemistry? A biomolecule, obviously it is a chemical compound, but interesting thing is it is found in living organism. Until now, all the different types of molecules that we have discussed, it is not that it is found in living organism, but here in this case they are naturally found in living organisms. What type of elements are mainly present? These include chemicals that are composed of mainly carbon, hydrogen, oxygen. So carbon, hydrogen, obviously these will be present because this is basically organic molecules, but other heteroatoms, oxygen, nitrogen, sulfur, phosphorus, that may be also present. They are the building blocks of life. They have different types of purpose, that is the reactions that we get from them. They have different types of purpose in the living organism, and important functions are performed by them. So here you can see some pictures for different biomolecules, that is examples: carbohydrates, protein, lipids, nucleic acids. Okay, now these biomolecules, they are used as fuel to provide the necessary energy for the different types of functions that are occurring in living system, in addition to biological function. So not only just biological function, it is also used to provide necessary energy, that is the required energy for living organism. And the field of studying about this chemistry, that is the chemical reactions that we can expect from this type of molecules, that is biomolecules, there is a different branch of chemistry which is biochemistry. Fine. So as if it is combination of two terms, biology and chemistry, whatever reactions are occurring in the living organism, comprising of all these biomolecules, that field of chemistry is known as biochemistry. Here we have this flow chart. So when we talk about biomolecules, carbohydrates, lipid, protein, nucleic acid, and for example under carbohydrate we have all these different molecules. So from this picture it is clear. Similarly under protein we have amino acid, peptide bond. So one by one we will see all these important 3 to 4 molecules. The first discussion will be related to carbohydrate. So what is carbohydrate? Just look at the name carefully. Carbohydrate. So as we are saying carbo, obviously carbon will be present, and they are the most abundant organic compounds in every living organism. And then there is also hydrate. Now hydrate, this is coming from water. It is related to water. That is why hydrate. So when it is water, obviously hydrogen and oxygen will be there. So here they are considered as hydrate of carbon, as if these molecules are hydrate of carbon. So carbon will be associated with some water molecules. So look at this formula, which is general formula Cn, n number of carbon present, which is associated with n number of water molecules. So that is why you can consider as if they are hydrates of carbon, and here containing hydrogen and oxygen in the same ratio as in water. So in water we know, if we consider the atomic ratio of hydrogen and oxygen is 2:1, 2 hydrogen, 1 oxygen. So same ratio you can also find here. So that is why they are hydrate of carbon. Now if we consider what types of groups are actually present, they are actually polyhydroxyaldehyde. Now when I'm saying polyhydroxy, that means several Wedge groups will be present. Wedge group will be present, and aldehyde, that means CHO group will also be there. So these are the groups that are present, or it may be keto group also. So in some molecules you can find keto group, in some molecules you can find CHO group, and Wedge will always be there. Fine. And so as a definition you can say they are polyhydroxyaldehyde or polyhydroxyketone, or it may be directly they are not polyhydroxyaldehyde or ketone, but they are producing compounds like this after hydrolysis. So both definitions are fine. So it may be that directly they are polyhydroxyaldehyde or polyhydroxyketones, or it may be that the after hydrolysis of these biomolecules, the final molecules that you are getting, that may be polyhydroxyaldehyde or ketone. But general formula is like this. Now, one important thing that you have to remember, though we are saying that this is the general formula, but not necessarily all the compounds that will fit into this formula, you have to consider them as carbohydrate. No, that may not be so. You can find so many other molecules which will match with this formula, but not necessarily it will be carbohydrate molecule. So that we have to be careful. Here we have some examples. Glucose, very important, and why it is written monosaccharide in the bracket we will understand after some time. Then sucrose, disaccharide written in the bracket, and starch, polysaccharide written in the bracket. So basically when we'll discuss classification, you will see that three broad categories. One is monosaccharide, disaccharide, and polysaccharide. So under each of these category you can see three examples, important examples: glucose for monosaccharide, sucrose for disaccharide, and starch for polysaccharide. Carbohydrates are synthesized, that is natural synthesis if you consider in the living organism. They are synthesized by the green leaves during photosynthesis process. Now this process is actually very complex process, but we are not going into the details. Important thing, in a simple way, what we can say, that this process is actually, it occurs under the presence of sunlight. So sunlight is actually providing the energy which is required for the production of carbohydrate. And what is the starting material? Starting material is carbon dioxide and water. So these two are the starting materials. In presence of solar energy or sunlight, we are getting glucose and oxygen. So if you look at the product side, we are getting not only glucose, which is actually the carbohydrate, but we are also getting oxygen. Now, if you consider the reverse reaction, the forward reaction, which is shown with the green colored arrow, that is actually occurring in plants, in green leaves. But if you consider the reverse reaction, now that is basically occurring, that is in cell. Animal, you have to consider now cellular respiration. So now there is respiration process, and in this respiration process, glucose is again converted to, in presence of oxygen, it is converted to carbon dioxide and water. And in this case, ATP and heat, these two will be required. Okay, so this is the reverse process. But if you want to know that how glucose is produced, that is basically by photosynthesis. And when glucose is decomposed in presence of oxygen, that is actually cellular respiration. Fine. Structure and classification of carbohydrates. So as you have seen in the previous slide, three main categories: monosaccharide, oligosaccharide. Actually there is disaccharide, but disaccharide is also one type of oligosaccharide. So better you use the term oligosaccharide, not disaccharide, because that is the broader term. Now this classification we are doing based on, it is based on what factor, that is important to know, because there may be different types of classification. But the important point for classification, which is based on what type of product you are getting after hydrolysis. Depending on that, the classification that you are doing, that we are going to discuss. So now we are having three types. Number one, mono. Number two, oligo. Disaccharide is just an example of under oligo. Disaccharide is there, and this is number three. So what is monosaccharide? So when it is monosaccharide, as it is mono, that means further hydrolysis is not possible. It is actually the simplest molecule that is possible. And if you consider oligo and poly, basically from these two categories you can get after hydrolysis monosaccharide. But monosaccharide, as it is the simplest molecule, you cannot further break it. So you cannot hydrolyze it to simpler molecule, because this is itself is the simple molecule. Now the next category, oligosaccharide. In this case, hydrolysis possible. Under hydrolysis, you can get 2 to 9 monosaccharide. Obviously, it has to be starting from 2, because if it is 1, then it is still monosaccharide, right? So at least 2 monosaccharide you must get from oligosaccharide. Otherwise, it will not be, it will be the definition of monosaccharide. Okay, so 2 to 9 monosaccharide you can get. But if it is more than 9, suppose you are getting 10 or 11 monosaccharide, now it will be under polysaccharide. So the first category, hydrolysis not possible. Second category, 2 to 9 monosaccharide molecules you are getting after hydrolysis. And if it is more than 9, now it is polysaccharide. So here it is mentioned, they generally yield a large number of monosaccharide units. So you can consider it as if monosaccharide is the monomer and oligo and polysaccharide, they are the polymer, because the small unit that you are getting from oligo and polysaccharide, they are actually the monosaccharide. Now if we see this flow chart, carbohydrate, simple and complex, and under complex mainly we have polysaccharide because it has to be under complex heading because you are getting large number of monosaccharide, so that is why it is complex, and the structure will be very large, that is expected. But when it is simple, you can consider mono and oligo, and under oligo, depending on after hydrolysis how many molecules you are getting, depending on that you can further do have some classification, which is disaccharide. That means in this case after hydrolysis you will get 2 monosaccharide, that is di. Trisaccharide you will get 3 monosaccharide. Tetrasaccharide, that means you will get 4, and in this way other names are also possible. So this classification, it is based on hydrolysis, right? But sometimes you will see other types of classifications are also there. That is reducing and non-reducing sugar. Now why we are saying sugar? To answer this, I can also say that there is third type of classification possible, where some carbohydrate molecules we call sugar because they are sweet in taste, but some are non-sugar, they are not sweet in taste. Okay, so this is actually another classification that will be discussed in detail later, but right now just see, when I'm saying reducing sugar, that means obviously we are now focusing on sugar carbohydrate, that is those carbohydrate which are sweet in nature. There may be two types. Reducing, so reducing sugar means obviously they will show reducing property, but if it is not showing reducing property, then it will be non-reducing. But again I'm saying here we will just see the simple definition, that is reducing and non-reducing. Detailed discussion we'll do it later. So as it is reducing sugar, so there must be free aldehyde or ketonic group, and as we know, failing solution tolerance reagent, these reagents we have discussed in aldehyde ketone chapter. So they are basically oxidizing agent. That means if you want to know whether any compound can show reducing property or not, you can do this test. So if these two tests are positive, that means the compound can show reducing capacity. So these two are the examples. And if it is non-reducing, that means free aldehyde or ketonic group not present and no reduction possible. This is an example. But detailed structure, why it is showing reducing capacity, why the other one is not showing reducing capacity, that we will discuss later. Now mainly here we have seen the classification which is based on hydrolysis, that is whether you are getting how many monosaccharide. Depending on that we can have three categories: mono, oligo, poly. Now we will focus on monosaccharides mainly. They are further classified. Further classification we are now discussing, because if we do so in the previous slide, it will be very complex. So in this slide, you will see mainly the classification under monosaccharide heading. Further classification possible, and that is based on what type of functional group present. Because as I have said, there may be presence of CHO group. For this, you have to use the term aldose, or there may be presence of C double bond O, for which you have to use the term ketose. So this is one thing. Another point is the number of carbon present in the chain. So if it is 3 carbon, triose, actually it is plural form written, trioses. If it is 4 carbon in the chain, tetroses, and so on. Now this carbonyl group, if it is aldehyde, then the sugar is aldose. Again, see, sometimes it may be that instead of writing carbohydrate, sometimes you can see in books also, it is written sugar, because several carbohydrate molecules, they are sweet in taste, they are under sugar category. So if instead of the term carbohydrate, you are seeing the term sugar is used. Don't be confused. It is actually meaning the same thing. I mean, it is also meaning that it is carbohydrate. So if the carbohydrate molecule, it is aldehyde group, it is aldose. If it is carbonyl group, it is ketone. Carbonyl group, that will be, sorry, it will be ketones, not ketone. Sorry, it is actually fine. It is saying that if the carbonyl group is aldehyde, then it is aldose, then this statement is finished. The next statement, if the carbonyl group is ketone, then the sugar is ketose. Okay. Now the most common monosaccharide you will see maximum up to 8, that is starting from 3 up to 8. This will be the number of carbons present in the chain. Now we will see some, that is in this column, you can see different types of monosaccharide. In the first column, number of carbon atoms that are present in the chain, that is mentioned. So if it is 3 carbon, under that 2 possibilities may be there. It may be aldehyde group, it may be ketone. So it is aldehyde. We have to use the prefix aldo and the suffix is triose because it is 3 carbon. Similarly, if it is keto, under 3 carbon number, it will be keto, then triose. Now the next two, again 4 because for 4 it may be aldehyde, it may be ketone. So aldo keto, just like the previous two, but now it is tetrose. Similarly, you can also understand for 5 and for 6, aldo pentose, keto pentose, aldo hexose, keto hexose. So these are the different possibilities depending on how many carbon atoms are present and what type of functional group is present. These two things we have to be careful. Now look at these molecules: prefix, tri, tetra, etc. Suffix, ose. So these are actually, you can also consider it as a prefix, but the next term, that is also a kind of prefix, tri, tetra. And ose, that is actually the ending term, so that is why we are saying it is suffix. So suppose if it is aldose and if it is triose, that means there should be aldehyde group and the total number of carbon in the chain, that must be 3. And it is aldotriose, the first example. So see, this is the structure: CHO group, then CHOH, then CH2H. Remember in the first slide what we have mentioned, it will be either polyhydroxyaldehyde or polyhydroxyketone. So here you can see these two are polyhydroxy 2OH, but aldehyde group is also present. Now the next possibility, again we have CHO group, but now 1 carbon extra, so tetrose. So see, as if this part is increased, right? So it is aldotetrose, this one. Now again increase 1 carbon. So as if another HCOH part is increased, and now the name is aldopentose. Now the fourth example, it is in blue color because it is now keto group, it is not aldehyde group, and total number of carbon 6. And keto group, obviously this aldehyde, they are present in the terminal position, right? But keto group you cannot have in the terminal position, because if it is in terminal, it cannot be keto. Both sides there must be some carbon present. So that is why now it is the second carbon. And the name is keto hexose. I mean this one, last one. And again, suppose you have same number of carbon, that is 6, but now the group is aldehyde. So it will be aldohexose. And under each, the picture that you are seeing, these are the corresponding names. So these names you can also find it here. So the first molecule that you had seen, that is aldotriose, glyceraldehyde. So this is actually glyceraldehyde, and this is the simplest structure for carbohydrate possible. Then we have D-erythrose. So erythrose is aldotetrose, this one. Now why there is mention of D, we will come to this point later. And then we have D-ribose, which is 5 carbon aldehyde, that means this one. D-fructose 6 carbon keto, this one. And aldohexose 6 carbon aldehyde, glucose. Fine. So in this way we can have different types of monosaccharide. Now what about oligo and polysaccharide? Now oligo and polysaccharide, that will be discussed in detail after finishing the details of monosaccharide. But here we will just have some simple idea and example. So see here, oligosaccharide, we have two examples. One is disaccharide, trisaccharide. Now disaccharide means these three examples that you are seeing, if you hydrolyze these molecules, that is if you hydrolyze maltose, lactose, then what you will get? You will get this combination of monosaccharide. And see, in each case, it is actually, it is not CT, it is actually two monosaccharide. So as you are getting two monosaccharide unit after hydrolysis, so that is why the name is disaccharide. Similarly, if you are getting three monosaccharide, obviously now it will be trisaccharide, and the corresponding example is raffinose. Fine. In this way you can have tetrasaccharide, but mostly disaccharides we will deal with. So here you can see the picture of raffinose. This is the first unit, which is galactose, then second unit glucose, and then fructose. So these three are monosaccharide: glucose, fructose, galactose. So you are getting total three monosaccharide. That is why it is under the category trisaccharide. So if you do hydrolysis with raffinose, you are getting three monosaccharide, and that is why it is trisaccharide. Similarly, if we do hydrolysis for maltose, we will get two monomer, that is two monosaccharide glucose. That is why it is under disaccharide. And what about polysaccharide? Sorry, this is not this poly. Under polysaccharide, now as we know, it will produce large number of monosaccharide, greater than 9. So these three are mainly important, and all these, how many monomers we are getting, that is not mentioned, but in each case the monosaccharide that you will get as monomer, in each case it is glucose. Though in each case it is you are getting glucose, don't think that they are same molecule. Obviously they are not same because of their different arrangement, and we will deal with it later after finishing details of monosaccharide. Configuration of carbohydrate. Now we have not seen, that is some pictures we have seen, and there what is the important thing, that there will be some OH group, and there will be either CH or C double bond O, that is ketone. Now when you have, just remember that structure, the carbon with which OH is attached, I am talking about that OH which is secondary alcohol, you will see mostly they are chiral center. Chiral center means all the groups that are attached to carbon, all of them will be different. So almost all carbohydrates, they are optically active, that is chiral, because of the presence of at least one or more than one chiral carbon. Now there is a projection formula that we use for carbohydrate molecule, which is known as Fischer. So Fischer has devised a projection formula especially for carbohydrates, and where you will see one of the two enantiomeric form of glyceraldehyde. Remember the first structure that we have seen in this slide, I'm talking about this one. This is glyceraldehyde. So this is 3 carbon and there are 2 OH group. This OH is primary, next one is secondary, and this is chiral center, right? Because in this carbon all the 4 groups are different. CHO group, H, OH, CH2H. So this carbon is chiral. That means enantiomeric forms possible. So that is actually just mentioned here, and this Fischer projection that we are going to see, that is actually based on this glyceraldehyde structure. Now what it actually means, look at this structure, both are glyceraldehyde, but this is D-glyceraldehyde, and if you take its mirror image, it is L-glyceraldehyde. So 2 forms are there, one is D, another one is L. Now how to know which one is D, another, and which one is L? That depends on when you write the molecule in Fischer projection by keeping the CH group at the top position and CH2H in the bottom position. Under this condition, if OH is on the right hand side, now it will be D. If it is in the left hand side, this L. But remember, you have to keep CHO group at the top position. Otherwise, this rule is not applicable. Now, keeping this basic structure same. Suppose in this part, just focus on this part where I am coloring. Here, with this carbon, directly CHO group is attached. But suppose with this chiral carbon, these two are chiral carbon, the CHO group is not directly attached. In between CHO group and the chiral carbon, there may be some other CHOH groups like this. So here, this part is same as glyceraldehyde, same. There is no difference. But now, in between CHO and this, we have some extra CHOH group. Okay, but as the bottom chiral carbon, it is just like glyceraldehyde, right? So that is why the sugar molecules or carbohydrate molecules, which are, having larger number of carbon. In this case, glyceraldehyde, you have only 3 carbon. Very simple. But suppose you have 5, 6 like this. In that case, it will be considered as D if the last chiral carbon, I'm talking about this carbon, if the last chiral carbon having same configuration just like D-glyceraldehyde, then that carbohydrate molecule will also be considered as D and the other one L. Now, actual example, this is the general structure possible. So many different possibilities are there because in between CHO group and the chiral said that you can have 3 carbon, you can have 4 carbon, different possibilities are there. Now, if you consider D-glucose and L-glucose, in between CHO and this part, in between CHO and this part, you have total 3 CHOH, right? Though there are different direction for weight. Sometimes OH is in the right, sometimes it is in the left, but that is not important. You just have to focus on the last chiral carbon. So you have to focus on this last chiral carbon. If it is matching with D-glyceraldehyde, the name will be D-glucose, and there may be any other name. In this case, it is glucose molecule, so that is why I'm saying it is D-glucose. And if it is matching with L-glyceride, it will be L-glucose. Rest of the part, because these are also chiral center, but we are interested when you are doing the configuration or you are suggesting the name D or L, you just have to focus on the last chiral carbon, not just any other carbon. Fine. So in this way, we decide whether it is D or L. Now, not just D and L. Carbohydrates are usually named with 2 types of prefix. One you have already seen, which is D and L, but sometimes you will see after D, this type of signs are also written. Sometimes plus, sometimes minus. Now don't think that D is always plus or L is always negative. No, D may be plus, D may be negative, L may be plus, L may be negative. Now when it is D and L, that we have already just learned, right? And carbohydrates are assigned the notation DLL by comparing the configuration of the carbon that is attached to CH2H group. Remember the last chiral carbon with that of glyceraldehyde. So comparing glyceraldehyde structure, we understand whether it will be D or L. But how to know plus minus? Okay, before going to the point plus minus, first look at these structures. Here we have in total 3 carbon chain, and this is D-glyceraldehyde. Gradually, we are increasing in this position some CHOH. So see, one CHOH is now added, and total number of carbon present in chain is 4. And this is erythros. But is it D-erythros or L-erythros? How you will understand? For that, you just have to focus on the last chiral carbon. So as it is matching with D-glyceraldehyde, this part is fixed. See, in all the molecules, this part is fixed. So that is why whether it is glucose, ribose, erythros, irrespective of that, it is always D. Okay, only we are increasing the in-between CHOH group. So in this case, it is 4. Now it is total 5 carbon chain, then 6 carbon chain. Fine. Similarly, if it is L-glyceraldehyde, keeping this part fixed, all these molecules, if you look at this part, it is fixed. There is no change. So as long as this part is fixed, all will be L because OH group, it is in the left hand side. So you can remember it like this, L for left. and we are just increasing number of carbon in the chain. Here it is 3, 4, then 5, then 6. So whatever is the direction of OH for the other chiral carbon, that you don't have to think about. Fine. Only the last chiral center, that is the carbon which is attached to CH2OH2, primary alcohol. That chiral center is important if we want to do nomenclature D or L. Now, what is this sign? Plus sign is used if it is dextrorotatory, minus sign is used if it is levorotatory. Now, what is dextrorotatory? Dextrorotatory, it is those carbohydrate molecules that will rotate the plane polarized light in clockwise direction. Now, plane polarized light it is a special type of light. It is not normal white light. Plane polarized light, it is there will be only one wavelength plane. It is a specially made light. Fine. So, if there is any chiral molecule, normally they rotate plane polarized light. And we know carbohydrate molecules are chiral, so it will rotate either clockwise or anti-clockwise. So if the molecules are rotating clockwise, it will be dextrorotatory or plus sign, and if it is if it is doing anti-clockwise rotation for the plane polarized light, it will be levorotatory. Now when it is dextrorotatory, it will always be plus. Now don't think this D is for capital D. No, this dextrorotatory means simply plus, and this plus it may be for D as well as it may be for L, both possible. And levorotatory, this L is not for capital L, no. Otherwise, this will not be mentioned here. Remember, levorotatory means it is negative, and it may be D negative or L negative. Both possibilities are there. But mainly not sign. Those signs sometimes you can see, because unless you know whether it rotates clockwise or anti-clockwise for the plane polarized light, it is not possible for you to predict whether plus or negative. So this type of sign, it will mostly not be given to you according to the syllabus, but D and L, that you can easily understand. So how we will know? You have to write the structure, or if the structure is given, you just have to check whether this CHO group it is at the top position or not. Under that condition, that is CH2H will be below bottom and CHO will be up position. Under that condition, if the last chiral center is matching with the configuration of D-glyceraldehyde, then it will be capital D, and the other possibility is capital A. Okay, so you have to understand that how to decide whether D or L. Sign that we cannot predict from the structure. For that, we have to do the actual experiment. Fine, but capital D and L, that you can do it yourself. Preparation methods for glucose molecule, because we are discussing basically monosaccharide, and under monosaccharide there may be different types of monosaccharide. See, in the previous slide you can have all these examples, erythrose, ribose, glucose, all are basically monosaccharide, but mostly glucose is discussed. So that is why we will see how we can prepare glucose. Glucose occurs freely in nature, and also it can be in the combined form. It is a simple sugar, because it is monosaccharide after all, no complexity is there. It is serving the major energy source for us. Most important, most abundant sugar, or better you can say most important, most abundant carbohydrate. It is present in honey, sweet fruit, grapes, mangoes, etc. And also we know that human blood also contains some amount of glucose, which is around 100 milligram per deciliter volume of blood. When it is in combined form, because in the first point it is mentioned it can remain freely or combined form. Now when I'm saying combined form, it is basically present in sucrose, starch, cellulose. Remember, these starch, cellulose, these are the examples of polysaccharide. That means when you do hydrolysis, you can get different monosaccharide, and this monosaccharide is glucose. That means it is in the combined form, that is a monomer form as if. So this is the method, which is laboratory method, or you can say small scale method, where we use sucrose as the starting material. Now sucrose will be, it is disaccharide, so obviously we will get two monosaccharide after hydrolysis, and it falls under oligosaccharide. So when it is boiled with dilute H2SO4 in alcoholic solution, it undergoes hydrolysis and give glucose and fructose. So these two are the monomers that you are getting, and both are basically monosaccharide. Glucose is monosaccharide, fructose is also monosaccharide. Though both are having same formula, but if you write it in Fisher projection, you will understand that bonding connectivity is different. Fine. If we want to produce in large scale, then we have to do the second method, which is starting from starch. Now glucose is produced commercially by this method. We are doing hydrolysis of starch, high temperature under pressure, and this is starch molecule C6H10O5 whole N number of water molecules, temperature 398 Kelvin, almost 400 Kelvin, pressure is very high, see 2 to 3 atmosphere, right? So we are getting N number of glucose, right? So from this type of formula, we are getting N number of monomer unit, or you can say monosaccharide, which is glucose. So this is the large scale method, commercial method, and the first method is small scale method or laboratory method. So these two methods are used for preparation of glucose. Now we will focus on glucose structure. It is aldohexose. Now when I am saying aldohexose, that means CHO group will be there, and 6 carbon chain hexose, 6 carbon chain will be there. Obviously it is optically active molecule. How many chiral centers are there? 4 asymmetric carbons are there. That is the carbons where the surrounding groups, 4 groups will be different. Now its solution is dextrorotatory. So that is why another name for glucose we say is dextrose. And remember, dextrorotatory, it is related to sign, so it will be plus sign. So this is the structure for glucose. See what we have written before glucose, it is D, because the last chiral center that is matching with D-glyceride configuration. And it is plus because it is dextrorotatory. So it will rotate clockwise, plane polarized light. So there are total 4 asymmetric carbon, that is 2, 3, 4 and 5. So these 4 carbons, they are actually asymmetric carbons, but other 2, they are not. Here with carbon you have 2 same groups H, so these cannot be chiral carbon. Fine. Now here you can see directly the structure, but how to derive this structure, that is actually based on some evidence. Suppose you have glucose molecule but you do not know its structure. So how to prove that this is actually the structure? For that we have to do some experiment. So this is the proposed structure for D plus glucose. Remember, simply saying glucose is not enough. You have to mention whether it is, at least you have to mention whether it is D or L. Otherwise the last configuration that may differ. Okay. So D plus glucose, which was derived based on some evidence. So these evidences are basically some experiment. Evidence number 1. If we do elemental analysis by mass spectrometry, which is basically done to understand how many different number of elements are actually present. So you can get the idea about the molecular formula of glucose. So from this experiment, it is observed that this is the molecular formula. But this is very basic information, because you just know that in each molecule there is 6 carbon, 12 hydrogen, 6 oxygen. But what is their bonding connectivity? What type of groups are present? That is not clear from. So this is a basic initial information that you can get from elemental analysis. We are just checking what are the different elements present. So 3 elements are present: carbon, hydrogen, oxygen, and their atomic ratio also we can understand. Evidence number 2. If you do reduction by using concentrated hydroiodic acid and red colored phosphorus. Remember, there may be different types of phosphorus: red, black, white, but you have to take red phosphorus HI combination, because this combination it is actually reducing it. Temperature 373 Kelvin. So if you maintain this, what you will get from glucose, you will get mixture of N-hexane molecule. N-hexane means simple hydrocarbon linear chain of 6 carbon, or you may get another molecule, though that is the minor product, 2-iodohexane. So hexane and its derivative 2-iodohexane. So if you are getting N-hexane as major, and you may not get this 2-iodohexane also, because sometimes you may get, sometimes you may not. So that is, it is minor product, but N-hexane you will always get. Now from this information, what conclusion we can draw. We can draw the conclusion. It indicates that 6 carbon chain is present. 6 carbon atoms are bonded in a linear fashion. That conclusion we can draw. So glucose, we don't know the structure, but after reaction you are getting these two products. From this we can draw the conclusion. It is linear chain of 6 carbon. Simple N hexane. N means it is linear. No branching is there. Evidence number 2. Evidence number 3. Now we will do reaction with hydroxylamine, that means NH2H. And another reaction is we'll do reaction with HCl. Now we know if there is any carbonyl group, whether aldehyde or keto, and under that condition, if you are using NH2OH, N2 hydrogen OH. So this is one reaction, NH2OH reaction with keto group. Another one is keto group in presence of Cn minus nucleophile, which is generated from HCl. So in the first case, what happens if aldehyde group is present? This O and H together it will be removed in the form of water. So you will get C double bond N, and that is known as oxime. As you are getting it from glucose, it will be glucose oxime. And if it is Cn, now it attacks here. C double bond O is broken, so you get Cn attached to carbon, and the fate of oxygen, now it is OH. So see, just focus on this part. CHO is now converted to something like this. CHO, it is now converted to oxime. Now if you are getting this type of molecule, the first one is oxime, the second one is cyanohydrin, I mean the final product. It indicates that carbonyl group is present, because hydroxylamine and HCl, they can react only with carbonyl group, whether it is keto carbonyl or aldehyde carbonyl, but carbonyl must be present. Then only you can get this type of reaction. Fine. So this is the proof carbonyl group is present in glucose molecule. Evidence number 4. Glucose, if you treat it with some oxidizing agent, for example, if you are treating it with bromine water, this oxidizing agent, it will convert glucose to gluconic acid. So what is the actual change occurring here? There is change only in this part. CHO group is oxidized to CO2H group, that is carboxylic acid. So it proves that it is aldehyde group, because if you are getting something else, suppose if you are getting not carboxylic acid, you are getting some other group, because only if there is aldehyde group present, then only carboxylic acid generation is possible. But suppose instead of aldehyde, if you have some keto group here, this hydrogen, suppose if it is not hydrogen, if there is some other R group, so obviously now you have to break some CC bond, and it is not possible to get CO2H group like this. There will be mixture of some other products. So if you are getting simple CO2H group, it proves that here the carbonyl group is actually aldehyde carbonyl, not keto carbonyl. Another oxidizing agent, if you use, okay, let me first finish it. If you use oxidizing agent like bromine water, it suggests carbonyl group is aldehyde group and it occupies one end of the carbon chain. And the other oxidizing agent, suppose now you are using concentrated nitric acid. Now this is oxidizing agent but not very strong. But now you are using concentrated HNO2, which is very strong. And now what will happen? Not just this CHO group, but the other terminal position, which is primary alcohol, that will also be converted to CO2H. So now both sides you are getting CO2H, and the name is different. It is glucaric acid, not O, it is A. Another name for it, also called saccharic acid. So it suggests that the other end there is presence of primary aldehyde group. Okay. So from the previous reaction, that is evidence 3, you have seen there is one CHO group, not 2, because it is 1:1 reaction. Per CHO you need one NH2H, or per CHO you need one HCl. Now one CHO group is present. But in this case, when you are using strong oxidizing agent like concentrated HNO3, and if you are getting 2 CO2H per glucose molecule, that means the other end, it must be primary alcohol group, because one aldehyde group is already proved. Other terminal, if it is not primary alcohol group, you shouldn't get this CO2H, because if it is secondary alcohol, suppose you have secondary alcohol, so both sides there will be some group. Now under this condition, if you do oxidation for this secondary alcohol, you will get ketone. But if it is primary alcohol, I mean, if instead of R there is H, 2H basically, RCH2H, now it will be COOH. So it proves that the other terminal, there must be presence of primary alcohol group. Evidence number 4. 5. Again we will do some oxidation reaction. And what oxidizing agent we are using? We are using ammoniacal silver nitrate, which is nothing but Tollens reagent. Silver nitrate, AgNO3, and another reagent which is Fehling's reagent, which is used for doing Fehling test, where what we take copper sulfate solution under alkaline condition. So here the actual reagent is basically copper 2 plus R, which is acting as oxidizing agent, because it will be itself converted to copper single plus, that is, it is accepting electron, so it is oxidizing agent. So in the first reaction, Tollens test, as we are using silver nitrate, that means Ag plus ion is there. Now this Ag plus, if it is converted to metallic silver, that means it will accept electron, so it is also oxidizing. So Tollens test is positive if you are getting metallic silver. Fehling test is positive if you are getting cuprous oxide. Cuprous oxide means Cu2O is the formula where oxidation state of copper is +1, Cu2+. So if you are getting this type of product, it further confirms the presence of CHO, because only the presence of free CHO group can give positive Tollens test and Fehling test. So here we have taken this Fehling solution. Actually it is AgNO3, but as you have taken it in ammoniacal state, so that is why there is NH4OH. So ultimately this Ag plus, you can also write it like this, as if it is AgNH3 whole 2 single positive charge, which is balanced by HO minus. Okay, so AgNH3 silver amine complex, in presence of ammonia, silver nitrate, it is changed to silver amine complex. But remember, actual focus should be on Ag plus, because ultimately Ag plus is converted to Ag0, which is metallic silver. And what is the fate of the molecule? Only change is in this part. CHO group, it is converted to gluconic acid. So gluconic acid means if only CHO group is CO2H. If you look at the previous structure, when you have used bromine water, basically you are getting gluconic acid. Only CHO group, in that part you are getting the change, it is converted to CO2H. Rest of the part no change. But when it is both CO2H, it is glucaric acid or saccharic acid. And in the second reaction, where actual reagent is CuSO4, but here it is written CuO, but important thing is it is actually source of Cu2 plus, and after reaction now it is Cu plus, that means it has accepted electron, it is oxidizing it. So the positive test means in Fehling solution, I mean, if you are getting red precipitate for Cu2O, because this molecule is red color, if you are getting this type of precipitate, it proves positive test. In this case also, only this part is changed to CO2H. Rest of the part there is no change. So it further proves that here free CHO group is present. Fine. Evidence number 6. If you treat glucose molecule with acetic anhydride, this is the structure of acetic anhydride. It suggests that is you are getting pentaacetate. Pentaacetate means if in your molecule there is some OH group, suppose you have any ROH group, like this. And now you are using acetic anhydride. What will be the fate of this molecule? In acetic anhydride, this part, COCH3 part, now it will be attached to the position of H. So I will not write H here. Instead, it will be COCH3. So try to understand the difference. H is now replaced by COCH3. And this is known as acetate. Now, if I am saying that you are getting pentacetate, what it means? It means there must be 5 OH group, right? Now, total number of carbon in the chain 6, that we know, and there is one CH group. That means among 6 carbon, if there is one CH group, rest of the 5 carbon, there will be 5 OH. And any type of alcohol, whether it is secondary alcohol or primary alcohol, in presence of acetic anhydride, you will always get this type of COCH3. Sometimes it is also written like this, ROAC. AC means COCH3, same. AC means COCH3. So pentaacetate, that is that we are getting, that means it proves the presence of 5 alcohol groups. So these 4 alcohols, they are secondary, and this OH is primary. But whatever, ultimately it is alcohol. So that is why we are getting in presence of acetic anhydride. Pyridine is also used because here byproduct is acetic acid. You are taking only this part, rest of the part is removed in the form of acetic acid. So this, to consume this byproduct, we use some basic reagent, which is pyridine. That is the purpose of using pyridine. But important thing is here, all these 4 OH and this another OH, 5 OH converted to OCOCH3. Look, now it is whole 4, but this part is, there is no change. So it proves the presence of 5 alcohol. Number 7. Glucose is stable compound. Fine. And dehydration is not occurring easily. Now suppose if you have any alcohol molecule and the same carbon is containing 2 OH group. There is any other bond, what bond, that is not important, but suppose you have 2 OH group attached to the same carbon. Now if this is the case, now dehydration becomes very easy. How? Like this. So now this OH bond is broken, there is C double bond O bond formation, and this OH is removed. So as if this OH and this this H and this OH is removed, and you get this type of dehydration product. So here water is removed. But if you have single OH in carbon and rest of the 3 other bonds, now dehydration obviously not possible. Next carbon may contain OH, but try to understand difference. Same carbon is not containing 2 OH. Individual carbon containing 2 OH. But if same carbon is containing 2 OH, this type of dehydration becomes very easy. But we have seen glucose is stable molecule, doesn't undergo dehydration, and it proves that not more than 1 OH group is bonded to a single carbon atom. And if there are total 5 OH group, that must be attached to 5 individual carbon atoms, 5 different carbon atoms, and the 6th carbon is aldehyde. Okay, got it? So don't think that this information, this observation, as it is not undergoing dehydration very easily, it proves that each OH must be attached to a single carbon. 2 OH attached to same carbon? No, that is not the case. In that case, dehydration will be very facile process, but you are not getting any dehydration, and it proves that all the 5 OH attached to 5 individual carbon, and the 6th carbon is containing aldehyde group. So keeping all these factors in mind, now this is actually the structure of glucose, right? So we normally denote the aldehyde carbon number 1, then 2, 3, 4, 5, And when it is glucose, then only the arrangement of these 4 OH like this. And the last chiral carbon, that is 5th carbon I'm talking about, this is always matching with glyceraldehyde. It is in the right hand side OH, that means it is D-glucose. Plus minus, that depends on the dextrorotatory or levorotatory. But ultimately this structure we can obtain from so many different evidences. Okay. Next important thing that we are going to discuss is that though from these 7 evidences we can reach the conclusion that this is the structure. Now this structure is open structure. You are not getting any ring. This is a linear chain, 6 carbon chain. One terminal there is CH, another terminal there is primary alcohol. I am talking about this. And in between these two there are 4 carbon, each carbon containing 1 H and 1 OH. But there are some reactions possible for glucose, or some observation is there from which it seems that this is not the only structure of glucose. I mean to say, it remains in some other forms also, and that is cyclic form. Because some reactions are there which indicates that cyclic form is the predominating form. But first, obviously it is important for us to understand the open structure, linear structure. Then only we can understand how the cyclic structure is possible. Fine. So that is why we have discussed so many things about this linear structure. It also remains in this form, but mostly remains in the cyclic. Fine. So next we'll see how we can reach the cyclic structure of glucose. It will not be finished into the class, just initial idea we can have. So Fisher identified that the open chain pentahydroxyl aldehyde structure that you have just seen, or the structure that we have just proved, did it completely explain its chemical behavior. So there are some chemical reactions of D-glucose which cannot be explained by its open chain structure. That means if you consider that no, this is the open chain structure, this is the only structure, then it cannot explain some observation or some chemical behavior. So what are those? Unlike simple aldehyde, glucose did not form crystalline bisulfide compound. Now if you have any aldehyde molecule, suppose you have any simple aldehyde molecule R C11OH. If you treat it with sodium bisulfite, NaHSO3. So this reagent, it is basically the source of the nucleophile HSO3 minus. Now this is a very large nucleophile. What it does, it can attack this carbon and there will be OH formation and you will get some adapt like this. This is very common for any simple aldehyde, but that you cannot obtain in case of glucose. So unlike simple aldehyde, glucose did not form crystalline bisulfite, and this molecule is crystalline. Fine. Then another test which is for CHO group mainly, shift test. That also we do not get from glucose. Then the third observation, if you take the pentaacetate derivative of glucose that was not oxidized by Tollens reagent or filling. Though we have seen Tollens reagent or filling test, but suppose in that case you have started the reaction with these two, in these two cases, you have started with actual glucose. But suppose first you have made the pentaacetate by using acetic anhydride, and then you are trying to do Tollens test or filling solution test. Now what we are seeing, we are not getting any reaction. So these are the some observation which we cannot explain if you consider that the open chain structure is the only structure. So next day we will see how we can reach from open chain structure to cyclic structure. We are at the end of the session. Thank you for listening.